Aldın mı? Mahkeme nasıl? Sen aldın daha taşınmadın. Gerçi öyle koşturuyorlardı yani. Kazanıyor. Hadi yaptı. Gerçi bunu danışmanın tam geçti be. Şirket de mi? Fabinho'da değil. Bulunan geriye göre güzel bir vermiş. Bir şey söyleyeceğim. Kılıfına beğendim de görmen gerek. Göster bize.
Good afternoon.
Good afternoon, Graham.
Hi, Stephen. How are you?
I am my usual self.
That's jolly good.
Yeah, I'm not sure what they want us to do. I've got a 10-minute presentation on engaging students in biomedical engineering with simple projects.
Sounds good. I'm just going to talk a bit about drug discovery and sustainability and environmental impact.
Excellent. So we'll have to go into the rainforest, into the woods to get our drugs now. Is that right?
Yes. No, this is kind of doing it the biological way, but with computers rather than large chemical plants and lots of plastic.
Excellent. Excellent. I'm really not sure what the format is. Apparently we're in part of a panel. Is that right?
Okay.
I don't know.
You don't know as much as I do.
Right. But anyway, at least the link works. So Arif's given us a link, so that's great.
Yeah, it's good.
Right. Well, I'll wait for it all to start.
Yep.
So I think they got paper presentations first, haven't they?
Yeah.
So we'll be on probably after about an hour, I suppose.
Yeah. Yeah.
Right, so around about
2 o'clock the session starts, doesn't it? So.
Yeah, and then I reckon we'll be on about 3 p.m, won't we?
Yeah. Yeah.
Okay, good. Well, that means I can do some thesis in the meantime.
Yes. I can put some expenses in, which is what's occupying most of my time at the minute.
Oh yeah, expensive. Got to do that, haven't we?
Yes.
Got it. Yeah. All right. Okay. Well, I'll catch up with you in a bit, and I'll wait out and see how things go.
Excellent. See you in a bit.
All right. Pleasure to see you. Cheers.
Artışı şu kadar olması. Bu sefer sen bazen çok Sen açmıyorsun. Ben seni tam seni sokma şansı olur. Değil de. O daha farklı gösteriyor. Authenticated. Faruk Hoca da acaba geçmiş olsun Allah. Günem mi? Evet. Kapat ediyorum. Upload ediyor, saving diyor. Karşılaştır. Lan disk kapan. Bak ya böyle. Abi nasıl yaptın? Nasıl
Welcome. Welcome, dear Stefan, and welcome. Welcome, dear Bell.
Hello, nice to see you.
Welcome. Welcome, dear Prof. Özcan Gündoğdu.
Hello there.
Hello.
Hello. Hiya.
Welcome, dear Ahmet. How are you, Ahmet? Welcome, dear Luis. Dear Luis, please open your microphone. Thank you very much.
Okay, sorry. Hello everybody. Nice to see you.
Okay, hello everybody.
Nice to meet you.
Nice to meet you.
Okay, thank you very
Hello
much.
from Mumbai.
Okay, let's start. Okay. Sorry. This is lecturer Mustafa Of from Kocaeli University and a member of Imascon organizing committee. Welcome again to the Imascon 2023 Spring Congress, where we held the 10th presentation and Congress. Imascon is a Congress where studies, different disciplines are presented. It's a great honor for us to see at such this event. And I would like to invite to dear Prof. Dr. Özcan Gündoğdu from Kocaeli University to moderate the session. Okay. Dear Özcan, thank you very much for support.
Thank you, Mustafa. I was asked to moderate this session at the last minute, so apologies if I make any mistakes. I met quite a number of you. I met Stephen recently. I met Louis a couple of years back, I suppose, back in Kocaeli. Hello, Dr. Ahmed Ali, to you too. Sometimes, okay, there are some words that become buzzword, and sort of like as the time goes by, they phase out. But sustainability seems to be staying on, and it is still being talked about for a long time. So today's theme is sustainability, and when it comes to sort of like to the word of sustainability, it means probably efficient use of resources. Basically, it sort of like bogs down to resource management, in my opinion. So when we look at the sustainability as a word, we come across this term: sustainability in banking, sustainability in environment, sustainability in sort of like various areas. So I haven't been provided what each of you is going to be talking about, but I presume sustainability will be sort of like in different perspectives. So let me start with Stephen Hughes. Hi, Stephen. Your mic is shut. Can you turn your microphone on, Stephen?
Now on.
Oh,
Hi there. Hi, everybody. I've prepared a very short presentation, and it's about sustainable technology in engineering education or medical engineering education. So just to introduce myself briefly, I'm a clinician. I'm an emergency medicine consultant working in the UK, and I teach at the medical school in Anglia Ruskin University. And what I'm going to do now is just share a little bit of the work that I've been doing with the medical students and with some biomedical engineering students here at ARU in Chelmsford. And I have just realized that I need to open. Better, you can't see that yet. Try again. Here we go. Right, can you see the presentation?
Yes, we can now.
That's fantastic. Right, okay, so thank you very much for inviting me to Imascon this year. This is my first time presenting at an international conference at all, let alone online. And I can tell you a little bit about my work. Now, I am very interested in respiratory rate monitoring devices. And these come in all shapes and sizes and all sorts of forms. But one of the simplest ways that you can get a signal from which you can measure the respiratory rate is by making a strain gauge. And they're simple, they're cheap to make, and they're quite accurate as well. And I find they make great student projects. They're brilliant for educating. So in the right panel here, you can see the strain gauge equation, not the best form, but certainly understandable to my students. And really getting medical students to do anything mathematical is very much an uphill task. They don't like it. But the strain gauge is understandable because it's physical, you can see it stretch, and you can see the measurement made, and they soon get the hang of it. Now, my work came about because I went to, get this to roll, I went to Jordan a few years ago, and I was shown this rather wonderful device here. This is a strain gauge made by Professor Ala Al-Halhouli and his colleagues at the German Georgian University in Mahaba. And what they did was they took some PDMS, poured it into a mold, and they put these rather splendid, let's get the pointer, these rather splendid liquid metal traces on there. It's silver nanoparticles. And they spent a lot of time learning how to embed their electrodes, and they came up with this device. It fits in the chest, and you can get a very accurate respiratory signal. Unit cost of that is around about $340 to $400 by the time you've purchased the silver ink nano printer and all the rest of it, and all these facilities. It has to be done in a dust-proof environment. It's actually was designed to be a resource for low resource environments, but actually the cost of this is really high. Well, here's my solution. If I can get it to go. So here are two versions of what I make. In the left panel, you can see a deluxe version. So I'll spend a bit of money on this. I've got a little bit of heat shrink on some silicone tubing, and I've got some recycled domestic copper conductor. And the tube is filled in with graphite in PVA glue. The cost of this is next to nothing. You can get PVA glue in an office worldwide, and would you believe it, you can turn your pencil shavings from the graphite core of your pencil into graphite particles, which serve the purpose very well. This is a sustainable strain gauge, and it costs, I've costed this out, this is my lab cost, 7 pence. And you get exactly the same signal with that as you do off the Jordanian device. The right-hand panel is what I'm working on. This is rather more durable. So these things last a good couple of years in use. And this is a rubber composite. You make a nice little wiggly trace on there just so that you reduce the strain across the whole length of the trace. You embed a couple of electrodes in it. You can fit that to the chest wall, and you get a fantastic respiratory signal. Cost of that is even less. The backing is a nitrile backing. In the UK, we use this as a disposable tourniquet for blood taking, but you could equally use bicycle rubber tire if you wanted to, or even party balloon. As long as it's flexible and as long as it's reasonably stretchy, it will work. And these things cost next to nothing, and they require very little by way of specialist equipment to make. Of course, my Jordanian friends weren't very pleased with this, but they humoured me anyway, and they didn't throw me out the country. They just carried on as if I wasn't there. Of course, you've got to demonstrate this. So here is some sustainable equipment. In the left panel, you can see a wooden testing rig. Here I'm hanging weights off a gauge, and then I've got it connected up to a data acquisition system. In this case, it's a good old-fashioned Arduino. Cost of this next to nothing. I built all of this out of my pocket money. The most expensive item in this was the motor that drives it. I got that off Amazon for just under £10. But the rest of this is all bits and pieces out of my junk box, and the wood came out of my roof because I had my house re-roofed. So I was left this great big pile of wood. I know I'll make a scientific instrument out of it. Now, this isn't so good. It looks terrible. So what I've done is, for the paper that we're going to be publishing, is I've actually taken this to my friend in the biomedical laboratory, and the biomechanics laboratory. She's got this testing rig where she can apply cyclical forces. And so I had a student spend a few weeks one summer just making strain gauges, and from this we could do very accurate measurement of the gauge factor, because the gauge factor for this is quite important. We showed that we could reproducibly make gauges depending on the mixture you use in our composite material between 2 and 20. So these are quite useful gauges. This is a good range for use on the chest wall. Now, we do have a little program here where we encourage people at school in their last year before they go to university, or the year before they do their A-levels, I should say, where they can do what we call STEM projects, science, technology, engineering and mathematics projects. And so for 2 weeks a year, we get young people into the laboratory and they do simple experiments. Now, I've just included my rig with these gauges. And last year's students, what they did on a project funded by the Nuffield Foundation was they embedded electrodes in various shapes into the composite mixture on a nitrile backing. And then what we've got here is a test rig. And what they were doing is pouring sand into a bucket. When the electrode pulled out, that was the applied force that you could put on. We wanted to know how we could embed our electrodes securely. And we tried 3 conformations. We had a loop, we had a straight piece of wire, and we had a helix. And we found that the helix was marginally better than the loop, and each of those were much better than the straight piece of wire. That pulled out fairly quickly with the applied force here. And with that, our young people went away very happily and decided to go and read engineering at university. So a bit of a success there for the engineers, not so much for the doctors. One of them decided he didn't want to do medicine, and that's great. So from these traces, we do get some fairly good. I'll just get this down. Some good traces. So this is a rubber glue composite. So this is bicycle tire cement. It's really cheap. It's £1.85 for a 100-gram tube. It's really cheap. You mix in a measured amount of graphite, and here I am with one of these on my chest wall looking at a trace, and here's the trace. They're really good and reproducible. With a bit of filtering, you can get rid of this artifact. That can be done with a very simple smoothing filter. This is almost a raw signal. So tiny bit of signal processing, and you get some great respiratory traces from which we can then automate the respiratory rate measurement. And all of this can be done on a simple laptop. And I've just realized that rather foolishly, I've put my laptop opening code. So I'm going to have to change the password for it after this. Silly me. Of course, whenever we test a device, we need a reference, and in this lab, we want to make it as cheap as possible. So for 10 pence, you can get a simple 10K thermistor. I've put a bit of heat shrink tubing on it just to make it nice and clean. I've put a couple of header pins on, and I've got some cheap cable here, and I've got a header pin arrangement. That's the reference device. That's a nasal thermistor. And ARU were very pleased to clear that as a device that we were happy to see used on staff and students. And the great thing about this is you can take this out and you can clean it, or you can just throw it away. Around COVID time, 10 pence a shot was thrown away, but we can actually clean these in a hypochlorite solution. And this particular brand of thermistor works very well. It's got a really low thermal mass, so it's really, really, really responsive. And you get some great signals from it. And here I am in my next slide. I've shown this at Kajali before. This is a comparison between the PVA strain gauge that I showed you earlier in this rubber tube and the nasal thermistor. And the thing you notice about this is that not only does the gauge work well with the reference, you know, you've got a waveform with each, but there's also a small phase difference. And this is the nice thing about these sustainable projects. One thing leads to another. We're not fighting with massive grants and things like that. We can do this sort of work on pocket money. And so I've got a couple of students investigating this phase difference. And what they found is, and this is preliminary results, is the phase difference changes between breaths. So here we show different breaths, okay? And the phase angle between the two changes, and then it resets after about 6 breaths. And this was quite a surprise, but we found that this was fairly reproducible. So again, you get this periodic dip. We've got longer traces which show much better sort of reproducibility of that resetting of the phase angle. And so once we've demonstrated this, we're hoping to get a device together to show that this is a function of vagal tone within the airways. And if we can do that, then we possibly got a means of screening for respiratory diseases using effortless spirometry. Now, all of this is highly sustainable. All of this is using cheap components. A lot of it's recycled materials. And from there, we go from a really simple strain gauge up to a rather more complex investigation concerning phase angles in the respiratory tree. So that's what I have on offer today. Thank you very much for listening. I am happy to take questions.
Thank you, Stephen, for a very nice talk. I suppose your talk is a very good example of sustainability in economics, sustainability in environment, and sustainability in science as well.
Well, it's sustainability in education as well. So this costs the medical school next to nothing to put on.
Fantastic. Thank you. Any questions? Okay, let me continue with Professor Ball. Graham, yeah. And can you put your microphone on? Yeah.
Okay. Good afternoon, everybody. I'm Professor Graham Ball from Anglia Ruskin, and my background is actually embedded strongly in sustainability because I did my first degree in applied biology, specializing in environmental biology, and I did my PhD, which was funded by the UN, in the modeling of plant-environment interactions with pollutants and making European scale models for pollutants based upon machine learning. So in 2000, I moved very much into more of a drug discovery, large-scale omics data modeling capacity. But I'm kind of going to merge those two strands today with the same sustainability focus, because what I'm presenting is a way to identify potential biological drug targets using machine learning as a much more sustainable method than conventional high throughput screening for drugs. So I'll just share my screen. So my title is In Silico Drug Target Discovery using machine learning. And this is a project that was funded by the UK government in lockdown to look at alternative non-lab based methods for drug target discovery. So in this space, there are a number of challenges. Drug discovery is a time-consuming process. It costs billions of dollars, and most drugs are recognized to have a 95% failure rate. In other words, only 5% get through to clinical trial. Our knowledge, however, of biological processes is relatively limited. So most of the drug discoveries are based upon the literature-based knowledge we have. And a lot of people are analyzing and mining the literature corpus. That is all of the papers ever written in order to try and find new drug targets. The problem with that is it gives a very non-specific view of biology. It's only the things that have been published will come up as potential targets. So what we're trying to do here is to use machine learning in a very different way to walk through molecular pathways, which are commonly used as drug targets, to fill the gaps and then identify drivers of those pathways as biological drug targets. So there are a number of challenges. Most of biology previously has just looked at fold change rather than the information content of genes. Biology is very non-linear, but often very linear and parametric methods have been used. There were big problems with false discovery because routinely we're looking at tens of thousands of genes in an individual's profile. And we have this problem called the curse of dimensionality, where the things we find can often be masked by the sheer volume of things we're looking at. So in this presentation, I'm going to use the P53 pathway, which is an important pathway in cancer, particularly in breast cancer, as a target. And this pathway is commonly mutated, and if it's the gene is mutated, there's an increased, more aggressive tumor. The P53 protects against cancer if it's not mutated. And P53 is related to uncontrolled cell growth, related to proliferation of cells, apoptosis, the bursting of cells when they die, DNA repair, and inhibition of angiogenesis. So it's a good and fertile ground for drug and biomarker discovery. So in this study, we used two data sets. The first one was Metabric, which I'm not going to talk about here, but the other one was the Cancer Genome Atlas, which had 1,197 breast cancer cases. And we specifically selected triple negative. That's ER, PR, and HER2 negative breast cancer cases. So just to illustrate some of what we're doing here, we're using neural networks, and these are a simplified model of a human biological network, although we're getting more and more complicated with the advent of and development of deep learning approaches. We're using these for classification, for regression, parameterization. So in a sense, we're looking at a model of human learning. Now, this is a very sparse model, but through deep learning, now we have many, many connections. We can weave in lots of functionality. What we're doing, in fact, is using a swarm of sparse models, which we create an ensemble with, which simulates a deep model, which we find is actually more efficient than just throwing a deep model in the first place. So just to look at what we are doing here, this is a representative population here represented by a well-known English sweet called a jelly baby. And let's say, some of these individuals have one phenotype and other individuals have a different phenotype. So we can profile that population with transcriptomics, with RNA-seq more recently. And then we can use our neural network to extract or pick the most important genes from that model or from that data to make a model that allows us to predict individuals who have phenotype A or phenotype B, and thus get a stratified population. So we're interrogating the omics data with machine learning. So we actually commercialized this back in 2010, and I'm also the CSO of a company called Intelligent Omics. And we've got a number of different approaches which we've developed on the back of that, one called Distiller, where we look at enrichment of markers, one called Pathway Miner, where we apply Distiller by walking through a pathway to find new features and fill gaps in the pathway. And the third one, which is Driver, which is a network inference-based approach, where we look at interactions between genes or between gene products to identify the most influential feature in the system. So that's more of a systems biology approach. So fundamentally, our approach is based upon a distribution of errors. And from that distribution of errors, we can repeat multiple analyses and examine the enriched lists, and then look for things that come up again and again and again, despite us perturbing that model by randomizing it. So if something comes up again and again and again, it's unlikely to be a false discovery. So we have this workflow where we get molecular data, we randomize the data into different splits, we train models on that data, we look for concordance, and then we take those results to a network inference approach. So in this study, we used that approach applied to the p53 pathway. So every green box in this diagram is a molecular target. And what we're looking for is the molecules that interact or associate with those targets. So to simplify that and illustrate it, let's say we have a hypothetical pathway. Molecule X links to molecule Y, which links to molecule Z. Molecule X links to molecule B, which links to molecule C, and molecule X links to molecule A. What we do is we mine each one of those targets to look at how genes in the whole transcriptome for a disease interact with each one of these. We then enrich that based upon the Distiller processes. So we find the things that are stably interacting and most strongly interacting. And then we look for associations between those. So the red dots here are effectively new features, new molecular features that are filling the gaps in the pathway. Because the pathways are generated based upon lots of little pairwise molecular experiments, this is taking a systematic view at a high level of the disease across the whole transcriptome. So these are things that may have been missed by conventional simple experiments. Once we have that pathway list, then we take that into inference modeling. So the point here is biology is not just a list of molecules. Biology is how things are interacting with other things, how they exist in a network or a cascade or a pathway. So what we're doing here is modeling that to understand how things interact with one another. So the way we do that is we make a model. Let's say we have 16 genes. We use 15 to predict one. So that's our first target. We parameterize that model. We then move on to the next target, train it and parameterize that, and then repeat that for everything. So everything is predicting everything else in a swarm of models. That gives us an interaction matrix. And that interaction matrix we can apply graph theory to. We can visualize it in Cytoscape. So this is an example of that. This is a real result for 100 transcripts. You can see the complexity here. And when we apply this to the p53 pathway, we get and filter it further, we get a number of hubs, strongly central features, which are driving the p53 pathway. Now, what's interesting is those drivers from other studies where we've knocked them out using CRISPR-Cas, where we've created transgenic plants with the targets from this, where we've used siRNA to silence those targets, they change phenotype. So the things which are central in these networks are the strong phenotypic drivers of the biology. And from that basis, it makes these very good drug targets, because what we want to do is hit the biology that has an influence. Otherwise, if we hit things that are off of the center, all that happens is the biological system will adapt. Biology is very good at working around molecular changes to a system. It's an evolutionary process by which we survive challenge from chemical influence. So we end up with these targets. We can then quantify that network. So we deconstruct the network with graph theory. And these are the top results here. What's quite interesting is our second most influential feature is a p53 apoptosis inducing protein. So some of the key drivers are the things we'd expect. But we also have some other novel, and unfortunately these are blanked out because these are currently being pursued as patent targets. But we have other drivers which are new, but more influential on the pathway than p53 itself. So these are new drug targets that we can potentially take forward. So we've validated a number of these. This is one example in the Lancet Oncology paper where we found drivers of proliferation called SPAG5. That was validated by siRNA knockout. It's been validated by immunohistochemistry. We've created a monoclonal antibody to it. That's very currently being pursued as a diagnostic. But once we have these targets, what we can also do is take these into databases. So one database, for example, is the Drug Genome Interaction Database, DGIDB. And I've taken TP73 here, and you can see here that we've got a reasonable hit, tautarotene, as a potential drug that would interfere with TP73. What we can also do is we can take that target into Chembl, and that's a much more rich ground to find new drug targets. So by looking at the things that are known to interact with given gene targets, we can find small molecules that are potential drugs. And so we get to the point where we have small molecules targeting the biology, which is actionable because it has influence. And those small molecules are shown to have a strong effect. Here, for example, in the order of 400 nanomolar concentrations. But we've got to this point without even touching any wet lab experimentation. It's all done in silico. So as a part of our Innovate UK project, we assessed the environmental impact of our method against the conventional for discovering a drug, and looked at how much effort would be needed to get to the point where we had, which was basically chemical targets which are ready to be taken into in vivo study. So the first impact was wastewater. Well, we are using electricity. There was no impact of wastewater, whereas a conventional high throughput screen where you're looking at multiple targets and also to get to the point of success has been estimated based upon cost of between 2,000 and 5,000 litres of water. In terms of single-use plastic, we use no single-use plastic, whereas conventional high-throughput screen because of pipettes, because of vials, because of growth vessels. They estimate is between 0.5 and 2 tons of plastic. Energy use, well, we have one PC, which is running at 750 to 1 kilowatt. Whereas if you look at all the energy use in terms of keeping vessels running robots, running heating, then to a high throughput scheme is around 35 megawatts. And from these, we've also calculated the carbon footprint, and our carbon footprint is of the order of kilograms, whereas the high throughput screen is the order of tons. And the last point to make is in terms of chemical things, chemicals entering the environment. Again, our approach has nil chemicals entering the environment, whereas a high throughput screen is applying thousands and thousands of chemical compounds to a cell line or the cell lines. So those have to be passed through some sort of sewage filtration. And from a typical high throughput screen, you're looking at between 3,000 and 5,000 chemicals being sent to waste. Now, those may be incinerated, those may be filtered, those may be drawn out by wastewater treatment, but nevertheless, they're still entering an environment. So this is potentially an alternative, more sustainable approach. Did we meet the challenges I stated? Well, drug discovery is time consuming. Well, we de-risked the time scale. We actually found our targets in a matter of 4 to 6 weeks rather than 12 years, which is a common estimate to get to a biological target from hypothesis. We overcame the limitation of understanding of biological processes by filling in gaps in the pathways. We build knowledge graphs based upon de novo findings from evidence, not based on the literature. We can take our models a stage further and look at the same pathway in healthy tissues, in off-target tissues. So we build the same model, but in, for example, healthy breast or liver, kidney, lung, skin, blood, and make sure that our targets are not driving the biology in those tissues. And our environmental impacts were hugely reduced. We estimated a 99.5% reduction at least in carbon footprint for our method. So this work has been funded and supported by a number of people. Intelligent Omics, there's Rob Grundy, Dimitris Kapsoulis, Dalia Mahasi. Work at Nottingham University Hospitals and Nottingham Trent University and the University of Nottingham. And the work here has been funded by Innovate UK, which is UK innovation funding body. Thank you very much. I'd be happy to answer any questions.
Thank you, Graham, for an excellent presentation. Any questions? Well, we have 2 excellent examples of sustainability. What about sustainability in human resources? Does that go into your remit, Graham?
Well, given that most CROs use robots to do the high throughput screening, and we employ people where we assign 3 tasks as a company to do the work, and it's not completely automated, and there is a human interpretation. We're not the kind of AI that's replacing people. We're augmenting the capabilities of people. So we're not creating driverless vehicles, which create a lack of jobs. One thing I would say is, I live in Nottingham, and Nottingham has a movement, or had a movement in Victorian times called the Luddites, and the Luddites went around smashing up weaving looms because they saw machines were taking over the jobs of people. And this is very at the forefront of my mind because I don't want to replace jobs. I want to create economies where people use machine learning as a tool.
That's correct. Correct. Thank you. Thanks very much.
Thank you. Thank you for the opportunity to present.
No, thank you for joining us. Can I continue with Dr. Ahmed Ali, if he's ready?
Yes, thank you. Sir, am I audible?
Hello, Dr. Ali.
Yeah.
Yes, we can hear you. We can see you.
So I will just make my work here. Okay, so I assume that my PPT is visible to everyone.
Fantastic. We can see the presentation now as well.
Yeah. So my work will be totally different from what my predecessor have presented. I'll be talking about a different aspect wherein the molecular mechanism of diabetes involved, and the process which I'm going to talk about is a fundamental process in the development of diabetes and its secondary complications. So the process is known as glycation. So I'll briefly describe the process itself, and then I'll be coming to a wonderful cyanobacterial organism, Spirulina platensis, which has been used since centuries as food and feed. And in recent times, the health potentials of this organism has been utilized. And one of the most wonderful thing about this organism is its high protein and nutrient, especially minerals content. So glycation is a process which is a non-enzymatic, quite different from glycosylation, which we have been reading in the textbooks, where the proteins are modified with the carbohydrates during the synthesis process in endoplasmic reticulum and Golgi bodies. So here there is an interaction between the sugar and the free amino group of other biomolecules, and the interaction is exclusively between the carbonyl group of the sugar and the amino groups. So this process keeps on happening in the body because there are sugars in the body and there are proteins. But the problem is realized, it becomes a problem when there is a condition that is called as hyperglycemia. So it's a very slow process because it's a non-enzymatic and covalent process. So it takes several weeks to happen in the body. So mostly those proteins which have longer half-life in the body are affected. So it can be divided into two stages, the early and late stage. The significance of dividing this is physiological as well as therapeutic, because once these products they cross the early stage, there is very less chances of reversing the formation of glycation products. And these products, when they reach into the late stage, they are collectively called as advanced glycation products, AGs in short, and they have the potential of altering the structures of the proteins like hemoglobin, albumin, induce DNA damage, and cause active oxidative stress. So one of the clear examples which I have shown here is glycated hemoglobin, and all of us are aware that now diabetologists rely on HbA1c for getting a history of hyperglycemic condition for that particular person. It gives an idea about the diabetic condition or hyperglycemic condition for last 3-4 months, keeping in mind the half-life, shelf life of the hemoglobin. So what happens? Hemoglobin gets modified by the same mechanism in which the amino groups or amino acid which have additional amino group like lysine and arginine, they get modified and it causes the structural alteration and finally the functional loss of the proteins like not only hemoglobin. Another example is albumin, which is more prone to this glycation because sugars and other things are more in the RBC. Collagen is another example, but most of the biomolecules get affected because the end result of uh, or the end mechanism of AGEs is through the generation of reactive oxygen species and reactive. So this small organism is the unicellular or multicellular cyanobacterial organism, which is photosynthetic and has been used as a food and feed supplement for centuries, and it has several additional benefits which environmentalists have been using for uh, bioremediation. So this has been an organism for great interest for physiologists and biochemists or people who are working on this. And another very interesting thing about this is has a very high protein content. So especially the regions which have the diets less rich in the protein, like rice is the staple food of the most of the countries. Rice is very low in the protein, so people are using this Spirulina platensis. And another is this in last COVID situation, we have seen people have reported this organism and its formulation for immunomodulation. So it's a wonderful organism. There are two main species, Spirulina platensis and maxima. So I have used. So what we have done, we have created an in vitro system in our lab where we have used protein as hemoglobin as a protein, and we have used different sugars because they differ from each other with respect to their activities. Like we have used a hexose sugar, and we have used its counterpart ketose sugar, and ribose is a pentose sugar. So we have used incubated the samples of glucose and hemoglobin, calling that as a glycation system, and then adding the formulation or preparation of esplatensis. So one of the hallmark of this glycation process, which was discovered by Louis Maillard in 1912, the process is also known as Browning or Maillard reaction, because the when the glycation happens, it causes the browning of the food, and that is what we have been seeing in the food industry because they are concerned about the quality of food. When you cook the food, when you at high degree, there is a browning that is because of the interaction between the glucose and proteins. But it also leads to accumulation of advanced glycation products, and that decreases or dot causes the uh, poor quality of the food. So we analyze what is the significance of. We can see that there is a significant amount of browning in all the systems, and uh, the presence of spirulina prohibited the formation or causing of browning by in a significant manner. Then we analyze the formation of HbA1c or fructosamine content, what we call it as. Then you can see that the maximum amount of glycated hemoglobin is formed in the case of ribose, 1.761 micromole per liter of deoxy formula. And we can we can show that the efficacy of extract in preventing the early glycation is more with the glucose system, maybe because of the reactivity of sugars like fructose and ribose, which are more reactive than the glucose. Then we measure the protein oxidation in terms of carbonyl content, and we can show that there is a significant amount of protein oxidation, which was suppressed by the spirulina extract. And we could show that the one which was maximally suppressed was and physiological significant also because glucose is the major sugar in our body. So spirulina extract could prevent the maximum amount of glycated hemoglobin formed because of the glucose. Then we measure the fluorescent edges. People use this as the marker. However, we cannot use this system because the only fluorescent edges and there are only few out of more than 200 edges which have been identified in the geological system. Only few are fluorescent, but still people use this as a marker for the analysis of advanced glycation products in the physiological body fluids. So we can show that there is a significant amount of AGS advanced glycation for the top bar, but the presence of spirulina extract prevented the formation of total AGS. Glycation induces aggregation also. So we use the thioflavin T and another aspect of fluorimetric method, very robust method for the analysis of protein aggregation. We could show that there is a suppression of aggregation by the extract also. So we can use this extract for the suppression of aggregation by any other process like thermal aggregation or other conditions which are there in the body. And this aggregation has been involved in the many of the neurodegenerative disorders. So we also analyze the effect of glycation on DNA, and then we use the spirulina extract to show whether there is suppression of this DNA damage, and this mechanism what is combinedly called as glycoxidative damage. So we can see here, this is the intact plasmid, this is the control plasmid DNA PBR322, and the lane 2 clearly shows that there is a cleavage of DNA which has caused the conversion of supercoiled plasmid DNA to the circular form, and then the presence of extract could reverse this damage or suppress this damage. So coming to conclusion, before that we have purified the phycocyanin, and we have shown in other study that most of this potential of spirulina extract can be attributed to the phycocyanin, which is the major pigment of this organism. And phycocyanin has been used to is being supplemented in the food as antioxidant and in the food industry also. So we coming to conclusion that aqueous extract of spirulina was found to be more effective in preventing glucose induced than ribose and fructose induced glycation. So as I mentioned that glucose is less reactive as compared to ribose and fructose. So glucose-induced glycation mediated aggregation was also severely suppressed in the presence of aqueous extract, and we could also show that glycoxidation, which is the result of the glycation adduct formation with the DNA as well as the generation of free radicals, and these two combinedly cause the damage of the DNA. So we could show that spirulina extract prevented the DNA damage. So I acknowledge my student work and the funding from various agencies. Thank you. I'll be more than happy to take a few questions.
Thank you, Dr. Ali, for a very nice presentation. Could I ask a question? I'm a member of a Turkish research funding body, and a project was submitted by one of the Turkish universities, and they were investigating the properties of Spirulina platensis on oxidative stress and DNA repair during chemotherapy and radiotherapy. Can you say a few things about it?
Yeah, as I mentioned that phycocyanin is one of the major pigment, and which has been shown to have lots of antioxidant potential. So that could be used as a to relieve the oxidative stress during many of this, and it has other antioxidants as well. Like it's rich in gamma fatty acids, which also show the antioxidant properties. So together, it's been in during the COVID time, many people have reported that it has immunomodulatory process activities. So altogether, they can be used for, and people have shown that anti-cancer, anti-proliferative activity of this.
Oh, excellent. I was surprised because it was the first time I came across it, and now you are saying that the aqueous form of it is more effective. Excellent, thank you. Any further questions?
And we have prepared this extract just with water and nothing else, no buffer and something like that, but just sonication. So, but
Okay.
however, it's a very difficult organism for genetic engineering. People have not been able to transform this organism. Yeah.
Okay. I have questions. Well, I have questions. Dr. Hughes, can you put your mic on, please?
Yes, silly me. Thank you very much. That very interesting presentation, Dr. Ali. Now, do rats with diabetes live longer if you've got a diabetic rat model? Do they live longer if you feed them this stuff, this spirulina extract? Is there any survival benefit if it's used?
Yeah, people have been, and there are formulations in the market where, I mean, it has been shown as a wonderful organism. So it can prevent the aging because of antioxidant properties, and anti-diabetic properties also have been actually realized through many clinical studies also. So, but the one problem with this organism is like it is in its structure. It allows the growth of many microbial organisms. So purity, maintaining the pure culture is very difficult, especially if you are going to use for therapeutic purposes.
Thank you very much.
Okay.
Thank you.
I have one more question from Professor Ball. Graham.
Given it's a plant system, I wonder if you've done any work to extract or run molecular profiling of the spirulina to identify what the active components could be. Many plant systems produce quite a lot of interesting phytoalexins, phytoalkaloids when they're under stress as well. So also, have you considered sort of changing the environment of growth to see whether that actually produces a more diverse set of compounds that we could look at in terms of their medical capabilities?
Yeah, thank you, Dr. Graham, for this very nice question. The problem with this organism is that it's very sensitive. It grows in alkaline system, and it grows between around 25 to 30 degrees centigrade. The moment you change it's very sensitive, it stops growing. So we could not find a way to change the environment or growth conditions to exploit the product. And one of the major phytoconstituent is phycocyanine. So there are it's rich in gamma fatty acids also, which has been like I mean people are proposing. So phytoconstituents are known, but the problem with this is that it's very sensitive to growth conditions. It grows in a special media, Xerox media, and if there is one component missing, it will stop growing.
Just one other thing. I'm in Mumbai at the end of July at IITB, so
yeah, that will be wonderful. Yeah,
please.
I'm working. Yeah, I'm working in University of Mumbai, and it's hardly half an hour drive, so we could catch up. Yeah.
Thank you.
Yeah, thank you.
Any further questions? Right. Thank you, Dr. Ali, for
thank
a very
you so
nice
much for your interest.
Thank you. Please do allow me to continue with Luis Rosero. Luis is familiar. Well, he's become one of us now. So, So please do continue with your presentation, please.
Okay. I think you are seeing my presentation.
Yes.
Yes. Okay. So maybe on this way, good afternoon. It's always a pleasure to be with friends, and I would like to thank the invitation. I am a mechanical engineering, so I'm talking about one of the new projects where I am involved. But first, I am part of the Polytechnic of Coimbra. It's an applied sciences university. Coimbra is in the middle of Portugal, between Lisbon and Porto. You are all welcome to visit us. And recently, I have new functions as an ombudsman of the Polytechnic. And the challenge to talk here is about sustainability. So I'm not working directly in the sustainability, but if we see the goals of development for a sustainable purposes, we can check that, for example, in my presentation, we are inside the health and well-being and the partnership for the goals. I mainly work in the biomechanics as a mechanical engineering, working with medical people and with some organizations connected to the hospitals. So this is one of the projects. Why I chose this? Because, okay, when we talk about good health and well-being, engineerings are always inside this, and must be in the development of a lot of different type of systems to improve these purposes. And particularly the mechanical engineerings can have a strong contributions to the type of competencies that we have. Also, and I hear this today from a colleague, higher education schools have an important role for the teaching purposes of the students, the new engineers, and working together for technology transfer. Also, as partnership, we have the questions about teamwork, very important, very essential, I can say, and multidisciplinary research and work, and of course, but our students working inside projects with companies and with institutions can be also very useful and inside the sustainability for teaching purposes. The problem that I am talking about is if we see the population aging is nowadays a growing phenomenon, and a very significant number unfortunately suffer from prolonged immobility syndrome. This can be due to loss of muscle mass, functional capacity reductions, and body systems. Particularly, we can talk about musculoskeletal, respiratory system, and cardiovascular. There are many others, but these, in this case, for these purposes, the World Health Organization say every day, please use regular physical activity for everybody. But in the case of bedridden patients, this is a problem. So how can we avoid this? Of course, rehabilitation therapy can avoid the immobilization's consequences, but fitted rehabilitation devices can improve the results. We can have better results with this. In my presentation, I talk about AbleFit is a new project. I just show you the product design, some development and preliminary applications. And this device can be used for active and passive rehabilitation exercises, and also for lower limbs and upper limbs. this is the main idea of the able fit. We can see a bed, a typical hospital bed, but can be a home bed for any person. And this is the first prototype of the system. It's designed for portability and the compactness for bedridden, but also can be applied for wheelchair users and can be used in hospital, but also in home training. The main movements associated with the able fit are exposed in this table. Can be for upper and lower limbs like abduction, adduction, internal rotation, flexion, extension, and so on, and applied for all of these type of members. In the concept of the able fit, there are two main things that have been developed: a linear system with a motor system control and the rotary system for this type of movements. This is connected to a microcontroller and an user interface, and the vital things can be monitored like heart rate, blood pressure, body temperature, frequency, etc. This is the prototype, the concept, linear and rotary movements. So as I talk, active or passive exercises, upper or lower limbs, and this is the base of the model, the interface for the patient, but also for the user, the rotating model and the linear model. Of course, when we talk about linear model in people with problems, for example, in the lower limbs, needing fitting for the foot, we can use, for example, these two types. The hand can be connected here, an handle, and if we want to use the foot, we have developed or implemented the clip-on pedal fitting will help, and it's easy to improve the movements. To get the quantification of the force applied for the monitoring inside this system, we have an embedded load cell, and the motor carriage of the linear driver, actuator driver, is shown to be very useful for this purpose. In the rotary part, the rotary actuator enables rotary movement control with force limiting. In this case, we also have load cells to control the force applied, to measure, for example, the force applied by both members. Here we can see an image, and a microcontroller received the set point definitions for each exercises, and all the sensors information is used to control both type of things like linear or rotary. The exercise data is forwarded to the tablet. Of course, what type of exercises? This project have inside the colleagues from the nursing school of Coimbra, and they have defined the exercises to be implemented with the system. So all the exercises have been well defined, and a classic loop takes care that the patient never exceeds its own physical limitations for security purposes. The force feedback is then activated by the quantification of the force obtained from the load cells. It's easy to implement, and instrumentation rotary and linear models can be easily used. The system have been, of course, projected to have the necessary strength and stiffness, mainly the structure for the system. And the clinics and the patient by its own can better adjust to the required force limits with the visual feedback that we have observed in the system. An Android-based tablet with a graphic interface gets the interaction with the patient and the clinical professional. Of course, we try to implement, nowadays it's very important, the gamification progress of the patient, and the personal data and the exercises are also get to a database, and the follow-up of the rehabilitation will be well defined in the preparation, but also in the future, so in the follow-up. The first experimental tests show that all the exercises can be implemented, can be configured and adjusted for the type of patients. The biofeedback data is collected and the exercises can be executed as prescribed. The graphic interface can be used to motivate the patient through the gamification, as I explaining, and the preliminary development of the biomechanical prototype is on the way to allow bedridden patients to carry on physical rehabilitation exercises, all the level of the upper and lower livers. And this is important both in active and passive mode. Just to finish, I'm going to show you a short video. If it, okay. The prototype was tested in the nursing school, and this is a project with financing from the European Union in a consortium with companies. So I hope that my presentation meets the objectives of the Congress. Teşekkürler. Thank you so much. And I finish my presentation.
Thank you, Luis. I think there is one question from Professor Özkan.
Okay. Thank you very much for your presentation, Professor Rosario. Muito obrigado. And thank is a great importance for my academic life. So I sent my greetings to my friends whom have done my many works with great success in ISAC, Institute Superior Engineering de Coimbra. And also in this, I would like to thank my colleagues in Coimbra. Another thanks, and I would also thank our professors from Anglia Ruskin University for their cooperation. Thank you so much. Thank you, Professor.
Okay.
Any questions? I have one question, Luis. Okay. What if a person is paralyzed neck down?
Sorry, can you repeat it?
I mean, a person cannot move any of his limbs or from neck down. He is completely paralyzed.
Yes.
Does this work for those patients?
It depends on the idea of the doctor. Maybe yes, maybe not. But any type of movement, if it will be supervised, can be helpful for the muscle. So the idea is, yes, to provide for long immobility, to provide exercises, even just the movement, muscular activity. So I think that even a lot, let's think about a person with in a post-stroke rehabilitation with one of the part of the body completely paralyzed. To check the movement for different time in the different times of the day will be useful, is what I have learning. So, but supervise it, not to encompass the limits of it, of the possibility. So it can be used for this type of persons also. But for example, for the main objective is for elder people and people in hospitals for a long time.
Okay, thank you. Any further questions? Right, thanks very much. Thank you.
If not, just before finishing, I would like, of course, to thank and say that Turkey is my second country and Kocaeli is my second home also. I feel always at home. Thank you so much. Thank you for your words, for the words of Arif. Arif, for me, it's a very important person. Also, it's a big pleasure to be with you and with friends.
No,
thank you so much, Professor. This is our pleasure also.
Thank you.
Thanks for joining us. The pleasure is all ours. Last but not least, can I invite Dr. Wakizaka for his presentation?
Şu an ses geliyor mu hocam?
Sorry.
Evet, alıyoruz.
Ses alıyor mu?
Evet, yes.
Yani evet, şimdi öncelikle değerli hocalar, özellikle burada bu değerli konferansa beni davet edip de sunum yapma fırsatını verdiğiniz için şükranlarımı sunmak istiyorum. Ve şimdi de özellikle davetli konuşmacı olarak katılmanın gururunu şu an canlı olarak hissetmekteyim.
Teşekkürler.
Şimdi burada değerli hocalar genellikle fen bilimler açısından kriz yönetimi ve sürdürülebilirlik konusunda sunum yapmıştır. Ve ben daha çok sosyal bilimler ve özellikle uluslararası ilişkiler açısından farklı bir perspektiften bilgileri hepinizle beraber paylaşmak istiyorum. Gerçi bugün anlatılacak şey genellikle geçen dönemlerdeki uluslararası örgütler gerisinde anlatılan şeyler de zaten burada anlatılacak konu yani kriz yönetimi ve dünya sisteminin değişiklikle ilgili konu olacaktır. Şimdi özellikle 19. yüzyıldan itibaren bugüne kadar dünya gelişme sürecinden bahsetmek isterse şöyle bir durum söz konusu. Zaten özellikle İkinci Dünya Savaşı'nın sonuna kadar zaten uluslararası siyasetteki aktör tek devlet vardı. Ve tabii bu durum aşağı yukarı Soğuk Savaşı'nın sonuna kadar devam etmiş ve tabii o döneme kadar özellikle dünya siyasetiyle ilgili yorum konusunda da yani realizm ve liberalizm gibi devlet odaklı teorilerin daha hakim olduğunu görebiliyoruz. Ama tabii özellikle Soğuk Savaş sona erdikten sonra şu durumda ortaya çıktı. Özellikle güney-kuzey uçurum sorunlar, uluslararası terörizm sorunlar, narkotik gibi uluslararası aşırı sorunların ortaya çıktığını görüyoruz ve tabii bu sorunlara karşı mevcut mevcut dünya sistemi bu tür sorunlara yeterli bir şekilde baş edemeyeceğini zaten anlaşmış durumda. Ve bunun aynı zamanda tabii mevcut teoriler, mesela realizm, klasik liberalizm ya da Marksizm gibi teorilerle tabii ki yorumlamak tabii ki yetersiz. Ve tabii bunun yerine devlet dışı aktörlere de odak veren daha bir esnek teorilerin hakim olmaya başladığını görüyoruz. Mesela inşacılık gibi teori var. Şimdi inşacılık teoriye baktığımızda güvenlik, ortak barış, dünya barış gibi konulardan daha ziyade devlet ya da bölgedeki kimlik, norm, ideoloji gibi unsurlara daha çok odaklanıldığını görüyoruz ve bu bağlamda tabii uluslararası örgütlerin yapıları ya da dünya sisteminin değişmeye müsait olduğunu üzerinde vurgu yapar. Aynı zamanda tabii Kopenhag'lı bundan da bahsetmekte fayda var. Zaten benim konuşmamın temeli daha çok bu teoriye odaklandığını vurgulamak isterim. Şimdi Kopenhag Okulu'na baktığımızda tabii güvenlikleştirme kavram çok önemli. Şimdi güvenlikleştirme kavramı nedir? Şimdi güvenlikleştirme kavramı anlatmak istersem bir siyasal iktidar bir sorunu güvenlik ile ilgili sorun olarak kamuoyuna sınarlar ve bu sorun güvenlik sorunu olarak karşımıza çıkar. Ve tabii özellikle sadece bir devlet değil, onun komşusundaki devletlere tabii ortak sorununu sunabiliyor. Ve tabii bu ortak algıya sahip olan güvenlik sorunu çerçeve etrafında tabii bölgedeki devletler toparlar ve ve buna dayalı bölge güvenliği kompleks gibi bir yapının ortaya çıktığına da vurgu yapıyor. Şimdi anlatabildim değil mi? Yani bir bölge ya da bölge devletler bir sorunu güvenlikle ilgili sorun olarak sınarlar ve bu ortak güvenlik sorunu etrafında bir ortak bölge oluşuyor. Ve buna bölge güvenlik kompleksi diyoruz. Ve şimdi bu tarihte baktığımızda özellikle günümüzde dünyada çeşitli sorunların çıktığını biliyoruz. Mesela özellikle 90'larda itibaren Kuzey-Güney ekonomik uçurumu sorunların hala tartışıldığını görüyoruz. Ve ayrıca tabii özellikle 2001'deki 11 Eylül saldırısından sonra daha çok uluslararası terörizm, bir de ayrıca narkotik, insan kaçakçılığı gibi çeşitli sınırı aşan sorunların gündemimizde olduğunu görüyoruz. Ve tabii bu tür sorunlar karşısında dünya yapısı nasıl değişti? Buna bakmak isterim. Şimdi tabii özellikle devletin ana aktör olduğu dönemde tabii özellikle tek çatı altındaki dünya sistem en azından işini görüyordu. Sonuçta Soğuk Savaş olsun, faşizm olsun, bunlar tabii dünya genelinde hep aynı sorunlarla karşılaştığını görüyoruz ve özellikle Soğuk Savaş döneminde dönemine kadar tabii devletin anahtör olduğu sorunlara tabii tek çatı altında sorunlar çözmeye çalışılabilirdi. Mesela Soğuk Savaş, dünyanın iki blok arasındaki bir çatışmaydı değil mi Soğuk Savaş? Bu yüzden daha çok bölgesel yapılanmalardan ziyade daha çok yani küresel yapı içinde çözmek en azından mümkün olduğunu görüyoruz. Ama tabii özellikle sınırı aşan sorunlar, mesela narkotik ticaret olsun, uluslararası terörizm olsun ya da yoksulluk ve açlık gibi sorunlar olsun. Tabii bu tür sorunlar her bölgeye göre farklı olabiliyor. Mesela Avrasya bölgesine baktığımızda en önemli sorun nedir? Uluslararası terörizm, ayrılıkçılık falan filan. Ama Güney Amerika'ya baktığımızda en önemli sorun narkotik sorun, ondan sonra yoksulluk gibi sorunlar var. Yani böylece artık bölgeye göre farklı sorunların mevcut olduğunu ve tabii farklı sorunların etrafında her bölgede ülkeler bir araya gelip bölgeselleşme sürecine doğru gittiğini görüyoruz. Ve tabii bu tür sınır aşırı sorunların karşısında artık tabii tek küresel çatının altında kriz yönetiminin artık yetersiz olduğunu görebiliyoruz. Ve tabii bu bağlamda özellikle farklı eski zamana göre farklı sınır aşan soruların karşısında daha çok dünyadaki yapılar tek küresel çatı altındaki yapılanmadan ziyade çeşitli bölgeye bölünen ve her bölge isimli çatı altında farklı dünya sisteminin oluşturduğunu görüyoruz. Mesela Amerika kıtasına baktığımızda daha çok Amerika Devletler Örgütü veya Mercosur gibi bölgesel çatı altında kendine özgü sorunlar çözmeye çalışırken Avrasya'ya baktığımızda tabii Şangay İşbirliği Örgüt ya da ASEAN gibi çerçevenin içinde ayrımcılık, yeni aşırıcılık ya da uluslararası terörizm gibi sorunları çözüme yönelik çerçeveleri oluşturduğunu görüyoruz. Ya da Afrika'ya baktığımızda daha farklı sorunlar etrafında bölge devletler bir araya gelerek bir bölge çatı altında sorunlar çözmeye gittiğini görüyoruz. Yani burada özellikle dünyada çeşitli krizler karşımıza çıkarken tabii burada vurgulamak istenen şu ki tabii dünyada çeşitli sınır aşan sorunlar ortaya çıktıkça artık mevcut uluslararası siyasetiyle ilgili mevcut anlayış ve tek çatı altındaki mevcut yapıların kriz yönetimi için yetersiz olduğunu anlaşılmaya başlamıştır. Ve tabii ondan sonra küresel tek çatılı sistemden ziyade dünya birkaç tane bölgeye bölünmeye doğru gitmekte ve bir bölge adlı çatı altında bölgeler bir araya gelerek kendine özgü kriz yönetimini geliştirmeye doğru gidiyoruz. Yani burada sonuç olarak vurgulamak istediğim şu ki özellikle dünyada çeşitli krizler ortaya çıkınca tek çatılı küresel yapılar bölünmeye doğru gitmiştir. Onun yerine dünya bölgeye, bölünmeye ve bu bölge altında kendine özgü kriz yönetimini geliştirmeye başladığını görüyoruz. Şimdi sunumu bitmişken sabrınız için teşekkür ederim.
Thank you, Dr. Wakizaka, for very good Turkish and very good roundup of impressive Turkish. Dr. Wakizaka talked about global security issues with the Copenhagen School of Interpretation and the global politics, the politics between the states, and with respect to terrorism, people smuggling, narcotics smuggling, and basically a global threat to people's security. So thank you. Thanks very much. Thank you.
Ben de teşekkür ederim Sayın Hocam.
Ben teşekkür ederim. Sağ olun. I think that concludes for today's session, and I would like to thank from the bottom of my heart to all participants who took their time and been here, gave their presentations. Thank you, Dr. Hughes. Thank you, Professor Ball. Thank you, Dr. Ahmed Ali, and thank you, Luis, and thank you, Dr. Wakizaka.
Çok teşekkür ederim.
Thank you to all our friends who have been here. And any last words, any of you?
Oh, thank you very much for hosting us and for an excellent set of presentations as well. Enjoyed the presentations of others on the call.
I certainly enjoyed all of them as well. Thank you. Okay, that's all for today, I suppose. Thanks again. Thank you.
Thank you. Have a good evening.
Okay. I hope to see you again.
Bye. Bye.
Place.
See you soon. Bye-bye.
Okay, bye-bye for now.